Good afternoon, students. Good afternoon, students. I'm very happy to welcome you for Zoom class. Today we are going to discuss about sexual reproduction. Before that, we have discussed asexual method of reproduction and vegetative reproduction, which we have discussed in previous class. Let us continue sexual reproduction. Sexual reproduction is a process by which fusion of male gamete and female gamete, that is called sexual reproduction. A fusion of male gamete and female gamete, which means male gamete and female gametes are fused to form zygote. That zygote develops into new organism. That is called sexual reproduction. So simply, by the fusion of male gamete and female gamete. Previously, the fusion of male gamete and female gamete is known as gametogenesis. But recently it is known as fertilization. Fertilization is a process by which male gamete and female gametes are fused to each other, right? That is called fertilization. Recently, before that, it is known as, previously it is called as gametogenesis, and recently it is known as fertilization. Now, fertilization is a process, it occurs in different stages in lower organism, right? In lower forms, the fertilization or gametogenesis that occurs in different stages. Fertilization, it is also known as gametogenesis. It occurs in three types. The first type is isogamy. Second one, anisogamy. Third one, oogamy. I mean, the fusion of male and female gamete to form fertilization. Fertilization occurs in different stages, in different categories, in different types. The first type is isogamy. Isogamy means the fusion of male and female gamete morphologically similar in size. So both male gamete and female gamete, which are morphologically similar in size and shape, that is called isogametes. Isogamy or isogamous means male and female gametes are similar in size, which are fused to form zygote, right? The zygote develops into new organism. That is the first method. And second method, anisogamy means both male and the symbol designated for male and the second symbol designated for female, both male gamete and female gamete, which are dissimilar morphologically and physiologically, which are dissimilar in nature. They are called anisogamy. That means fusion of male gamete in one side and fusion of female gamete in another side and shape, which are fused to form fertilization. Such a condition is said to be as the anisogamy. And third one, that is the oogamy. Oogamy is nothing but the male gamete is very smaller in size and the female gamete, which is very large, right? So male gamete is just very smaller in size and female gamete is very larger in size, which are fused to form fertilization. Such a condition is known as oogamous or oogamy type. Mostly, this iso mention gametes. Generally, the gametes are in different size and different shape. Talking about fusion of male gamete and female gamete, here I like to mention gametes or the unicellular structure which is either in the form of motility or non-motility. So simply gametes are divided into two types. The first type is a motile form and second one non-motile form. What is the difference between motile form and non-motile form? If you take a male gamete, now this is a male gamete or female gamete, either male or female gamete, in which there will be flagella or present. The flagella or present. The flagella or locomotory structure which helps the gamete to migrate from one place to another place. Such a condition is said to be as a motile form of gamete. Whereas non-motile form of gamete male, now this is a gamete. This is a gamete. It does not arise whether male gamete or female gamete. It does not arise flagella. The flagella is totally absent. That is called a non-motility or non-motile form of gamete, right? So there are two types of gametes which are involved in motility and non-motility. Motile form of gamete produces or that arises flagella. Flagella are essential for moving from one place to another place. Whereas non-motile form of gamete, it does not arise any other appendages or thread-like structures or flagella. Such a condition is said to be a non-motile form. Right. This is whether motility form of gamete or non-motility form of gamete which are fused to form fertilization. Right. Okay. This is a types of fertilization. Okay. What we are going to see in fertilization or in sexual reproduction by the fusion of male and female gamete. Right. Before going to deal these changes, these structural changes or these physiological changes in plants, we have to deal the types of structures of various plant part. The first one is the flower. Because a flower is when you see the flower, this is the most attractive part of the plant. It is a tool for, it is an inspiration tool for, particularly I can say for poets. It is a reproductive organ of the plant in which you can see there are mainly four parts of flower. The first one is, I'm talking about parts of flower. Because flower is a reproductive organ in which both male reproductive organ and the female reproductive organ are present. The male reproductive organ which are essential for the male reproductive organ is nothing but stamen is the male reproductive organ. The group of stamen is known as the androecium. The androecium and the gynoecium or the reproductive structure which are formed in a flower and which are protected by these reproductive organ of male and the female flower are protected by a non-essential part such as sepal and petal. A sepal, a group of sepal is called calyx. So calyx is the outermost line of the flower, outermost wall of the flower that covers corolla. Actually, corolla, a group of petal is called a corolla. So both the calyx and corolla are protective part of male reproductive organ and the female reproductive organ, right? So this is the structures of fertilization, right? So I like to mention once again to you. The flower, if you take flower consists of calyx, corolla, androecium, gynoecium, which are the main walls of a flower. The outermost layer is called calyx. Calyx is a green in color. It is the outermost part. that consists of number of sepal. This is a sepal region, and followed by corolla. Corolla is the attractive part of the flower, which is made simply corolla. Corolla is nothing but it's a group of petal that covers, that encloses reproductive organ, the male reproductive organ, that is androecium. And just inner to androecium, you can see the female reproductive organ, that is a gynoecium. The gynoecium which lies at the center, it is called as a female reproductive organ. So we are going to deal both male reproductive organ and the female reproductive organ, right? This is called the free fertilization structures and events, right? Okay, in which one we are going to say, first of all, free fertilization structure and events. I'd like to draw the structure of free fertilization structure. Free fertilization structure. Free fertilization structure and events. Structure and events. Right? What are free fertilization structure? Both male reproductive organ and the female reproductive organ. So the male reproductive organ part and female reproductive part. The female reproductive parts, it's nothing, it's a gynoecium. The gynoecium is a female reproductive part and the male reproductive part is androecium. Androecium is nothing, it's a group of stamen. Now this is a stamen. The structure is called androecium. Androecium is nothing, it's a male reproductive part that consists of anther, a second one followed by connective tissue, and third one, stalk of the anther, is called filament. Filament, it's nothing but it's stalk of the anther that carries out anther lobe. Both anther and the filaments are connected by a specialized tissue that is called the connective tissue. This is a structure of male reproductive part, I mean androecium. So today we are going to discuss about only androecium, the parts of androecium, and what are the stages are involved in the development of anther. Then what are the walls are formed in anther. Then what is the function of this anther, right? We are going to discuss one by one. First of all, let me take anther or androecium. When you take a cross section of the anther or stamen, you can see different stages, right? So first of all, let me take the formation of anther in plant. Actually, in plant, the anther primordium. Actually, anther, I'm talking about development of anther. I'm talking about development of anther. So now this is a anther primordium. Actually, anther primordium is an initial stage, right? The development of anther is initiated by this anther primordium, right? Anther primordium is nothing but there is a group of initial which is surrounded by epidermal cell. I'm talking about anther development or development of anther. I'm talking about the development of anther. Simply, the anther encloses a group of tissue, a mass of tissue that is called a homogeneous tissue. This homogeneous tissue is surrounded by epidermal region, right? And some layers of this homogeneous tissue gives rise to form the fertility cells that is called the hygroscopic layer or the hypodermal region. The hypodermal region of the cells the hypodermis, the cells of hypodermis gives rise to form archisporial cell. Actually, archisporial cells are formed here. Archisporial cell. This archisporia which initiates a mass of parenchymatous cell which is differentiated into on one side parietal cell, on another side sporogenous tissue or sporogenous cell. This archisporial cell, once again it differentiates and makes and pushes parietal cell towards outer, and the sporogenous cells are towards the inner side, right? After the formation of parietal cell and the sporogenous cell from the archisporial cell, it has to be developed. As it is, here the parietal cells are responsible for the formation of anther wall. We have discussed two types of tissue. They are parietal cell and sporogenous cell. I'm talking about this. The first thing, primary parietal cell. The parietal cell are responsible for the formation of anther walls such as endothelium, middle layer, and tapetum, right? So this epidermis is outer covering of anther, then followed by the second layer is endothelium. Before that, the epidermis is outer covering. When you take a cross section, it is nothing but when you take a cross section of anther, consists of stamen, anther lobe. This is the anther, and connective tissue. A stalk of the anther is called the filament. Right? The anther and the connective tissue filament. When you take a cross section of the anther, you can see 4 layers. The first layer is epidermis. The first layer is epidermis and followed by endothelium. Actually, endothelium is the second layer of anther. It lies just beneath the epidermal region, then followed by middle layer. The middle layer, it is the third wall of anther. It is also originated from the primary parietal cell, and the innermost layer of the wall is called tapedum. Actually, tapedum is the innermost wall, innermost layer of anther. So these structure you can see when you take a cross section of the anther, you can see 4 walls mainly. They are epidermis, endothelium, middle layer, tapedum. At the center of each and every lobe of anther, you can see a group of mass of tissue that is nothing but it's a sporogenous tissue. This sporogenous tissue are developed into microspore mother cell. MMC, microspore mother cell. This is nothing. It is capable of forming microspore, right? This is a development of anther wall and microspore mother cell. The formation of microspore by the process is called microsporogenesis. We are discussing about the different stages which are involved in the development of microspore. So this is the next stage in which the formation of microspore is called microsporogenesis. Microsporogenesis. What is microsporogenesis? Microsporogenesis is a process by which the formation of microspore. What are microspore? Microspores are a haploid condition which are derived from microspore mother cell. Actually, this is a microspore which is a haploid condition. It is derived from microspore mother cell, or MMC, microspore mother cell. It is responsible for formation of microspore. Right. So this microspore mother cell is derived from this microspore mother cell, which are derived from microsporogenesis. Simply I can say sporogenous tissue. So the last generation of sporogenous. The sporogenous is a tissue. The last generation of a sporogenous tissue directly may function as microspore mother cell. The microspore mother cell undergoes for meiotic division to form microspore. Actually, microspores are haploid in condition. All the microspores are sometimes union in nature. All the microspore held together. Simply, the microspore in sporangia or united or held together to form like a pollinia. The next stage is called as a pollinia. Right? This is one of the development of microspore. Generally, microspores are occurred in different stages, right? So always the microspores are union or unite together, right? All the microspores inside which are united together. So simply The microspore in Microsporangia to form as a pollinia or pollinium. Pollinium is a singular and pollinia is a plural form. Right? So the microspore in Microsporangia or union, right? It's to form as a pollinia, right? But talking about pollinia, this is a pollinia. The microspore in pollinia to form a clamp or clip-like structure that is called corpusculum. Corpusculum. Actually, corpusculum is nothing but a group of microspore in Sporangia. In Microsporangia are united together to form like a pollinia. All the pollinia to form like a clamp-like structure, clamp or cup-like structure that is called corpusculum. The corpusculum which arises, there is a thread-like structure that is called retinaculum. It is called retinaculum. It is called retinaculum. And the retinaculum is also again, it is connected with the pollinia. So the whole structure, it is represented like a inverted Y, that is called a translator. It is also called as translator. So the translator is nothing but the whole structure resembles to be like a inverted Y shape. That is called a translator, right? So this is simply something about structure of pollinia. So this pollinia is reported in case of Calotropis. I am talking about pollinium, or pollinium is a singular. Pollinium. A group of microsporins, Microsporangia held together, right? That is called a pollinium or union in nature. Example, Calotropis. Calotropis, they are the example of Oscillopitaceae family, and the Calotropis are the example of pollinia, right? Next we pass on to the development of when you take a cross section of the anther, just once again I'd like to remember the structure of anther. Now this is a structure of anther. We are discussing about mature anther. When you take a cross section of the anther, The anther shows the following parts: the cross section or the T is of mature anther that shows the outermost layer is the epidermis, which is made up of a single layer of parenchymatous cell without any intercellular spaces, but sometimes the outer covering is interpreted by the presence of, there is a minute opening that is called a stomium, right? So that is called a anther cavity. The anther cavity is created by the presence of stomium. Actually, the stomium is responsible for dehiscence and liberation of microspore from microsporangia. I mean, the liberation of pollen grain from anther lobe into outside, right? That is the role of anther cavity, right? The next one, endothecium. Endothecium is a second layer of mature anther, which is made up of very large elongated cells that lies just below the epidermal region. It is also responsible for dehiscence of anther and the development of middle layer. Actually, middle layer is a third layer which is made up of 2 to 3 layers of anther wall, right? It is also present just below the inner to endothecium. And finally, the fourth layer is a tapetum. Actually, tapetum is a fourth wall, fourth layer of a TSM mature anther that's responsible for development of microspore, which are found inside. Right? This is a microspore or the pollen grain. The pollen grains are developed from microspore. Before that, the microspores are haploid in condition. So this haploid condition of microspore, which gets nourishment from the tapetum, right? So this is a role of the tapetum. But tapetum generally it is of two types. They are secretory tapetum and invasive tapetum. Tapetum it is of two. They are secretory tapetum and invasive tapetum. Secretory tapetum and one more category, invasive tapetum. Actually, secretory tapetum is found just inner wall of the tapetal. This inner part of the anther wall, that is a middle layer, which are arranged in a manner, but whereas the invasive tapetum, the tapetal cells are arranged loosely, right? That is the major difference between secretory tapetum and the invasive tapetum. The tapetal cells are arranged in a manner in case of secretory, whereas in invasive, the tapetal cells are arranged loosely, right? That is called the invasive tapetum. The next we pass on the functions of tapetum. What are the functions of tapetum? The first point, tapetum which nourishes food material for the development of microspore. That means it provides food material for the development of microspore, which are grown inside of the anther lobe. Second one, it also contributes a sporopollenin. It also contributes sporopollenin. The sporopollenin is a material which is essential for the formation of outermost layer of the exine. I mean the pollen grain. The pollen grain is surrounded by two layers. One is the intine and the other one is the exine. The intine and the exine are contributed by the contribution of sporopollenin. Actually, sporopollenin is essential for protection of this pollen grain. One third point, then pollen kit also produced. Pollen kits also contributed by the presence of tapetal cell, right? And it is also responsible for exine and intine. It is also responsible for exine, rejection and reaction process. rejection and reaction. Rejection and reaction process. So this is a major functions of tapetum. I repeat once again. Tapetum is the innermost wall of the anther in mature anther. The first point, it provides the food material for the development of pollen grain, which are microspore inside of the anther. Second point, it also contributes to sporophyllin for the formation of the exide. Exide is the outermost layer of pollen grain. Third one, it also contributes to pollen kit, right? Next one, it also produces some proteins which are essential for the formation of rejection reaction process, right? For the process of rejection and reaction process, by which the pollen grain is easily attached on the stigmate region of radiation, right? So this is a major functions of tapetum. Next we pass on to if you take an anther, you can see anther cavity. And the first thing, andrecium consists of andrecium is nothing but a group of stamen is called a group of stamen. It's called andrecium. The andrecium, a group of stamen is called andrecium. Now this is a repeatedly I am drawing the structure of it is anther, and second one connective tissue, and third one filament. Right, anther we discussed, we have taken cross section of the anther, that we have discussed four walls of anther: epidermis, endothelium, middle layer, tapetum. Already we have discussed, there are four walls. The first one epidermis, and second one endothelium, and third one middle layer, and fourth one tapetum. Already we discussed. Next, anther cavity. Actually, anther cavity is preserved, which are filled with the microspore. Some anther cavity, I am drawing, just I point out here. Let me point out, this is a anther cavity. The anther cavity are nothing but they are the micropores which are distributed all over the anther lobe, which are filled with more amount of microspore. Actually, microspores are present. Microspores are filled with it, right? So the anther cavity. The anther cavity is in which you can say stomium. Now when you see the structure, now this is called a anther cavity. This is the anther cavity. This cavity is filled with more amount of anther cavity is filled with more amount of microspore, right? The next one, connective tissue. The connective tissue, this is a connective tissue. The connective tissue is a sterile. Connective tissue is always it is a sterile in nature, but that encloses the sterile condition of tissue, the mass of tissue that encloses vascular tissue. from the connective tissue to the development of microspore in anther cavity. Actually, connective tissue which provides the food material. So mineral water are given by only connective tissue because vascular tissue is present inside of the connective tissue. Even though connective tissue is a sterile condition, but it supplies water, mineral, the development of tapetal cell and the development of microspore and the microsporogion, right? So this is simply something about of male reproductive organ. So far we have discussed the male reproductive organ. What is male reproductive organ? Androgen is a male reproductive organ, right? In which anther lobe, if you take androgen, you can say this is a androgen. Androgen consists of stamen, connective tissue, filament, etc. Right? So anther, it's a bilobed in nature. When we take a cross section of the anther lobe, you can say there will be 4 cavities, right? And stalk of the anther is called a filament, right? So this is simply something about of male reproductive organ or male reproductive part of the plant. and what is flower, and what is the pre-structure and the events of fertilization, right? Could you understand? Students, am I audible? Swati, is this audible? People support 40 notes. There are 70 students. How can happen? Students, could you mention the walls of anther? Anyone, please? We have discussed 4 walls. They are endodermis, epidermis, endothelium, tapetum. Will you mention the walls of anther?
Yes. Talk loudly, Swati.
Epidermis.
Epidermis, very good, good.
Endothelium.
Very good. Second layer, that is second layer. Then
Middle layers.
middle layer, okay.
Tapetum.
Tapetum, very good. So this is a walls of ATS of mature anther, right?
Hello.
Okay, the next we person to. What are the different types of gametes that we have discussed? Could you mention, please, students? We have discussed 2 types of gamete. Will you mention the name, please?
Yes, tell us, Divyashri.
Please tell us the types of gametes.
Motile and non-motile, sir.
Okay, how did you differentiate motile and non-motile? Okay, students, please tell us, you mentioned very clearly motile form of gamete and non-motile form of gamete. What are the difference between each other? Students, I have drawn the diagrams very clearly. Just
Yes.
yes. Motile arises flagella.
Pleasant.
Okay, absence of flagella in non-motile, right? Very good. Right. Okay, what is called stomium? What did you understand about stomium? We have discussed the stomium and pollen grain. What do you understand about pollen grain? In the structure, when we take a cross section of the mature anther, that shows following parts: epidermis, endothelium, middle layer, and tapetum. Inside of the anther lobe, we can see some structure which are star-like structures. Could you mention their name, please? Students, anyone?
Abhishri, Swati, Tamil.
Right. Anyway, no problem. When you come to next class, make an assignment. Just I'd like to give some question to the topic. Draw the structure of TS of mature anther. That is the first question. And second one, write about the functions of tapetum. Right. We draw the structure of flower and label the parts. So there are 3 questions are given to you. Thank you. Thank you for joining.